All right, guys, this is homework number 1 for the second 6 weeks. It's going to be on DNA structure, and so your essential question is, what is the structure of DNA? So make sure you have your Cornell notes set up. So this would be your title right here, and then you have your essential question. Okay, so let's go ahead and get started. The first thing we need to talk about is, what is DNA? And DNA is described as the genetic material that determines our traits. So another word for traits are characteristics. So how we would describe people, whether you have brown hair or blue eyes or you're 6 feet tall, those are your traits, those are your characteristics. And DNA is the genetic material in all of our cells that determines those traits for each individual. DNA is passed on from one generation to the next. So for example, mom and dad pass on traits to you. So you get your genetic material from your parents, and then you will pass on your genetics and your genetic material to the following generation. So continuing on with what is DNA. DNA is a biomolecule. So it's a molecule that's essential for living things. And because it's a biomolecule, it's going to have a monomer and a polymer, just like our carbohydrates, lipids, and proteins. So the monomer is called a nucleotide. A nucleotide. Its polymer, okay, is a nucleic acid, which is also just the DNA strand itself. So this picture you're seeing here would be the DNA strand. That is its polymer. So let's begin talking about the monomer of DNA, the nucleotide. So this monomer is pretty complex compared to the other monomers we've looked at before. It's made of 3 components or 3 parts. The very first one is called the phosphate group. Right? Okay, so this right here is the phosphate group. A lot of times you're going to see it drawn as a circle. So if you will notice, we now have a new element. It is a P for phosphorus. So nucleotides have carbon, they have hydrogen, they have oxygen, they have nitrogen like our proteins, as you can see right here, and they also now have a phosphorus or a phosphate group. So that is something new that you're going to want to keep in mind for a nucleotide. Let's see. Okay, the next part is the sugar. The sugar. So the sugar in the DNA nucleotide is called deoxyribose, and that should be relatively easy to remember because DNA stands for deoxyribonucleic acid. So the D in DNA stands for this sugar, okay? And so the sugar is right here. Okay, you can tell that it is actually a carbohydrate because it's a sugar. So nucleotides actually have a carbohydrate in them, this deoxyribose sugar. The last part of a nucleotide is called the nitrogen base. Sorry, that's kind of light, but it's called the nitrogen base. And there are 4 bases in DNA. We have adenine, thymine, cytosine, and guanine. And you're going to need to know all of these, okay? So a lot of times we can refer to them as A, T, C, and G. But you need to know the names also just so that if you see them on a test or a quiz or in a reading passage or something like that, you're not totally thrown off by the fact that they're using the whole name. But we will refer to them in class a lot as A, T, C, and G. Okay, so in DNA, there are 4 bases, as we just talked about. So there's A, T, C, and G. These bases do something called base pairing, okay, base pairs. So that means each base is going to pair with another base, like a pair of shoes. They go together. So to start, adenine, or our A base, adenine, pairs with thymine. pairs with thymine. So if you look over here in this picture, okay, you can see adenine right here and thymine right here. And so adenine is green, thymine is pink in this diagram. So every time you see another adenine molecule, this green base, you see a pink one attached to it. And that's because wherever there's an adenine, there's going to be a thymine. So it's kind of like wherever there's a left shoe, there's a right shoe, right? The next one is that cytosine, the C base, always pairs with guanine, the G base. Okay, so again, if you come over here and look at this diagram, the guanine base is the blue one and cytosine is the white base. Okay, so every time you see the blue, it is paired, oh, up here it's orange. Okay, that's fine. But every time you see the blue, it's paired with a guanine over here. Okay, one of the memory tricks that I know I teach my kids to use is that cars go with garages, right? So a car goes in the garage, and apples grow on trees. So A and T go together, apples grow on trees, and then C and G pair together because cars go in garages. So if that helps you, feel free to use it. If it doesn't, just forget I said it. So the next thing that's important about DNA is how do we actually get these bases to pair together? And so DNA is held together with hydrogen bonds. So that just means that in between these bases, there are little hydrogen atoms that hold them together. And these hydrogen bonds are actually very weak. There's just a lot of them in our DNA that holds everything together. And it's important to keep that in mind, the fact that they're weak, when we start talking about DNA replication here in the next couple of weeks. Okay, so as we discussed, nucleotides are the monomer of DNA. And so our nucleotides are going to bond together to form the DNA strand, which is the polymer. So if you look down in this picture right here, it has boxed a nucleotide. So a nucleotide includes three parts: a phosphate, a sugar, and a base. And so the circle is what you see is the phosphate. And then this little, I guess, pentagon right there is the sugar, and then this rectangle, is the base. And so every single one of these is a nucleotide. And so they are going to pair together, they're going to connect together with those hydrogen bonds to form the whole strand of DNA, which is this up here. So one important characteristic of DNA is that the bases are bonded together anti-parallel to one another. So what this word means is we probably have heard the word parallel before, and parallel lines look like this, right? They run alongside of each other, but never actually touch. So anti-parallel just means that these lines are going to run parallel to one another. You can see that these two strands of DNA run parallel to one another. However, they're kind of upside down. So for example, if you look over here, okay, this nucleotide, the phosphate is on top, if you will, and then there's the sugar, and then there's the base. Over here, though, okay, we notice that the phosphate, the circle, is towards the bottom. The sugar is flipped around so that the base can pair together. Okay, if they weren't anti-parallel, these bases wouldn't be able to connect to one another. They would be flip-flopped. And so that's just something important to keep in mind. You might see it written as 5-3, and we'll get into what that means a little bit later. But just know for now that they are considered anti-parallel. They're flip-flopped from one another. And one of the last things that's important about DNA is that DNA forms a double helix when the nucleotides bond together. Sorry for my typo. So when the nucleotides all bond together, we get this double helix shape. So you can see over here, it kind of looks like a twisted ladder. Okay, so in the center are the the nitrogen bases, and on the outside, this right here, are the phosphate and the sugar. So we actually call that a phosphate sugar backbone. So you can see this picture also. You can see the AATs, Cs, and Gs bonded together. We call those the base pairs. And then this blue side is the phosphate sugar backbone. So that is made up of the phosphate and the sugar. And so when those bonds connect, when those base pairs bond together, they form this double helix shape. They automatically twist and form like a twisted ladder. And that's it for homework 1. So make sure you go back through this. If you can't answer the essential question, make sure you re-watch parts of the video to make sure you understand. And if you're still struggling to understand some things, feel free to ask a question tomorrow in class. Have a good one.